Dr. Cepko's current research is focused on the development and degeneration of the retina, as well as on the development of gene-agnostic therapeutic strategies. So Dr. Cepko, it is my pleasure to present the Friedenwald Award to you, so please come forward.
Are we done?
Okay, so now I'm pleased to introduce Dr. Timothy Cherry, Associate Professor at the University of Washington School of Medicine. Okay, there we go. So I was going to say I was otherwise not loud enough to be able to reach the back. So who's Associate Professor at the University of Washington School of Medicine's Department of Pediatrics and Associate Director of the Center for Developmental Biology and Regenerative Medicine at Seattle's Children's Research Institute. So he's going to tell us more about Dr. Cepko and all of her achievements, as well as introduce her lecture.
It's a tremendous honor to be able to introduce Connie for the Friedenwald Award lecture. I'm told I was chosen to do this introduction because I could be trusted to stay to the time limit. For those of you who know her work, Connie is, this award is richly deserved. Connie is truly a visionary among vision scientists. For those of us like myself who are lucky enough to train in Connie's lab, she's been a transformative mentor. Personally, one of my most meaningful memories of Connie is when I was getting ready to move across the country with my family to start my own lab in Seattle. At the time, I thought, this is truly a moment of profound uncertainty. Connie reassured me by saying, you know, Tim, there are many ways to succeed in science. I found that deeply reassuring because Connie had taught us through her own example how to carve our own research path and push visual science research in new directions. In preparing for this introduction, I reached out to as many of Connie's previous trainees as possible, and as you can imagine, the response was tremendous. I won't be able to share all of the responses that I received, but before we welcome Connie up onto the stage to give her lecture, I'd like for you to be able to hear in some of the words in the words of the trainees what Connie has meant to them as a mentor, as a scientist, as an innovator, and as a role model. So if you could please play the clip.
What's remarkable about Connie is she's a great mentor.
As a mentor, Connie supported me in unusual circumstances. I asked to join her lab shortly after she accepted a faculty position. That support made a big difference to me, allowed me to finish graduate school and start a career in research.
I'm grateful to Connie for her mentorship, which has influenced me deeply. She introduced me to the world of molecules and viruses. She's someone who has exceptional knowledge and thoughtfulness, and this coexists with genuine modesty and kindness.
Connie's positivity and resilience in the face of scientific challenges made me stronger and helped me persevere through some of the most difficult times in life and research.
Thank you for your support and mentorship over the years. You were always supportive and honest to me, something that's not always easy to do.
As a mentor, Connie puts people first. You always left the meeting excited and energized about your project.
As a mentor, Connie taught me that excellence in science goes hand in hand with generosity and integrity.
She taught me how to be a rigorous and fearless scientist. I witnessed how she focused on the science and put her trainees first, above all the others pushes and pulls of a leader in the field.
She was always so enthusiastic and eager to hear what my results were. I would come to our one-to-one meetings sometimes and be a little depressed that things weren't going as quickly as I hoped, and I would always be just inspired and eager to do the next experiment by the time I left her office.
Something I learned from Connie that I've held on to is that it's never too cold to bike to lab.
Something I've learned from Connie that I have held on to is her love for detail and the way of second questioning all the data that you get.
What's remarkable about Connie as a scientist and a mentor is how she embodies rigorous scientific excellence while remaining deeply humble and human.
What is remarkable about Connie is her passion for science. What really matters to her is always about the science itself, about asking the right questions and finding the correct answers.
Something that I learned from Connie is the importance in using precision when making descriptions. For example, I will never ever confuse a precursor from a progenitor from a stem cell ever again.
Connie has been a role model for me in how her genuine interest in a scientific question is always at the forefront of answering that question.
Has been a role model for me in rigorously performing good science while being extremely humble.
Connie has been a role model for me in countless ways. I'm truly grateful for the time and wisdom she has shared with me.
Connie has been a role model for me as a brilliant and confident scientist, a kind and caring mentor and mother, a fun and interesting person.
Connie has been a role model for me in being technically innovative and scientifically fearless.
What I learned from you is the importance of invention in scientific discovery. In our case, it was barcodes and retroviruses, but you've just invented one thing after another after another.
She's a leader in her field, a woman scientist, deeply respected for excellent and rigorous research, a scientist with remarkable breadth, applying multi-scale approaches to answer complex biological questions in the retina, from developing molecular tools to conducting multi-species comparative analysis. She's also a genuine and passionate scientist and mentor.
Yeah, hi Connie. Your award was long overdue for your numerous contributions to vision research.
Thank you very much, Connie. And congratulations.
Thank you so much for all you taught me and all I've learned from you.
Congratulations, Connie.
Thank you, Connie.
Thank you, Connie.
Congratulations, Connie.
Thank you, Connie.
Please join me in welcoming Dr. Connie Cepko for the Friedenwald Lecture.
That was amazing. Well, that was amazing. It's a little hard to go on now, but thank all of you. I have a lot of trainees sitting here. Thank you, Tim, for putting that together. I know it takes quite an effort to edit videos and compile all those things, and you did a beautiful job. Thank you so much. I also want to thank Anand Swaru for the nomination and the awards committee for giving me this. I'm really thrilled to be here. There's so many of you out there. Wow, look at all these people. This is amazing. And I want to thank all of my trainees, because I really wouldn't be here today without the work and the inspiration and all the great conversations and interactions I've had over the years. I've been really lucky to have a number of wonderful colleagues, trainees, and it's been great to work with all of them. It's really been a fantastic time to have such people to interact with each day. I just love it. I also want to thank my family. I have a few family members here, my husband, David Cardozo, and my daughter, Leah Cepko. I also wouldn't be here without their love and support. Just as an example of David, I think he's forgiven me for forgetting our anniversary last year, I hope. And Leah's amazing. I'm kind of clueless when it comes to a lot of cultural things. She gives me my clothes. She sends me playlists for Spotify. She sends me recipes and keeps me up on all of the latest foodie things. So I've had a wonderful family life, and it's really been very supportive and helped me carry on as I have as a scientist. I also want to thank all of you for being here. We're in a really tough time. It's really challenging. It's not going to be easy. But I think what we have to hold on to is science is still science. It really doesn't matter what the political climate is. Science is wonderful. It's true. Facts are facts. And we're really the privileged people who get to carry on trying to understand Mother Nature. And this is really a wonderful pursuit. We're very privileged and lucky to be able to do it. It's going to be hard to continue, but I think what we need to do is support each other. And I know it will get better, I just don't know when. Okay, on to science. Speaking of beautiful, this is one of the reasons I love working in the retina. It's a beautiful image by Sui Wang when she was a postdoc in the lab. It's a cross-section through a mouse retina, and it shows the beautiful lamination, which enables fairly simple cell type identification, just based on where a cell resides. It also shows a lot of beautiful colors, which are from electroporation of plasmids, which encode fluorescent proteins, and this has enabled us to study gene regulatory networks. This was invented basically by Takahiko Matsuda when he was in the lab. So today what I'm going to tell you about is our thoughts on how this structure develops. So I'll give you an introduction to retinal development, some of our old lineage experiments, a little bit about how we've tried to analyze what's going on in retinal progenitor cells. These are the cells that make, these are the mitotic cells that make all the retinal neurons. Also, a model we now have for retinal development. And then I will show you some pretty pictures of genes that we hope are patterning the high acuity areas, much like the fovea that you just heard about. So the retina starts from this simple neuroepithelial layer. This is a mouse embryonic day 10 section. And those greenish cells are all mitotic neuroepithelial cells that are dividing away to create a larger number of epithelial cells. These will give rise to all the retinal cells. In the very center, the first neurons will be born, and being born means going through a terminal S phase. And there's progressive waves of genesis of each retinal cell type from the center out to the periphery. And at the end, one ends up with about 120 cell types. We know this number mostly from single-cell RNA-seq, but we also have a lot of classical descriptions based on morphology and physiology. Now, the cells, the retinal progenitor cells, must use a strategy during development that solves several important questions. So first, there's diversity. I just mentioned 120 cell types. That's quite a task. And amazingly enough, these cell types have to be distributed equally throughout the retina. So when we have light hit various regions in our retina, we have local circuits to carry out computations that are useful to us, direction of motion, changes in intensity, et cetera. So for that to happen, each of these cell types has to be equally distributed throughout. This is a pretty picture of an on-alpha ganglion cell from the cat from Vesely. But all of the retinal cell types exhibit this so-called tiling. And there's an amazing difference in the frequency of the retinal cell types. So on the left, my left, your left, is an outline of a mouse retina drawn by Dick Maslin and colleagues. They were looking at wide-field amacrins. And those green processes are a single amacrin cell that covers about a quarter of the retina. There's about 16 types of these wide-field amacrins. They're very rare, only about 500 per retina, which is a frequency of about 0.005%. The other image shows rod photoreceptors. So they're the majority of our photoreceptor types, about 95% in us and about the same in a mouse. And in a mouse, they're 70% of all cells in the retina. So this means there's 5 logs of difference in the production of these different retinal cell types, and somehow the progenitor cells have to do that. And finally, you just heard about a beautiful area in our retina, one that we hold dear, the fovea, where we have our high acuity color vision. That has to be placed in a particular place during development, and it has a different repertoire of cell types, for example, no rods and all cones. So the retina has to have a strategy that allows it to do all of these things, and to do so fairly robustly. This is the order of genesis of the cell types, and by genesis I mean terminal S phase. This is data from a mouse, from Richard Young, and on the left is the embryonic stages, and on the right, the postnatal. And you can see that there's this nice order. But even at one time and place, more than one cell type can be born. For example, a single division can make a cone and a horizontal cell. Now, this is a very conserved order. All the species that have been studied, and this includes birds, mammals, amphibia, they all have this order. So we've wondered a lot about this order. Why is there this order? Perhaps it reflects the evolution of the retina. The earliest retina might have had a photoreceptor, an output neuron, and an interneuron. Those are the firstborn cells. And there's a little bit of evidence of this from hagfish. It also might be the order of addition of these other cell types, with Mueller glia being the remaining progenitor cells. In fact, they do express the progenitor genes, and they can serve as stem cells in some organisms. Now, when we started studying the retina, we noticed this, and we were very interested in the relationships of the mitotic cells in these different daughters. So we wanted to carry out lineage tracing experiments. For example, Mueller glial cells might have a glioblast progenitor. In fact, if you read textbooks, they talk about glioblasts, which are mitotic cells that just make glia. And we thought there might be a photoreceptor blast, because rods and cones are so specialized, maybe they have their own progenitor. So to look at these questions, we use retroviral vectors that I developed as a postdoc, and we deliver them in vivo, rats, mice, and chicks. We deliver them subretinally so that those viruses encounter the M phase cells on the apical surface of the dividing cells. And they integrate the viral DNA, and it can't spread to neighbors. It can only integrate into the infected cell. And it's replicated along with the genome and passed on to all the daughters. And we can look anytime later for evidence of the histological stain that the virus enables us to see. And you can see a clone on the right there. And one thing that was incredibly fortuitous is that sibling cells have a radial distribution. So if we put in a sparse amount of virus, we can find well-separated clones. We've done a lot of work to show they're clones. And it enabled us to quickly analyze what was going on in terms of progenitors. So this is Dave Turner, my first graduate student. You saw him on the video. This is Dave's experiment, our first lineage tracing. We have some from the rat and from the mouse here. On the left, those two panels are from postnatal day 0 rat, and they're fairly small clones, with rods, a Mueller glia, and a bipolar on the far left, which reminds me to tell you there's no glioblast. We've never seen a clone with just Mueller cells. On the far right, we have a clone initiated at embryonic day 14 in vivo. It's amazing that Dave was able to do that. And we have a lot more cells, so we have rods, Mueller glia, bipolars, amacrins, and ganglion cells. And the take-home message from all of this, from our work and that of others in frogs and fish, is that these progenitors are multi-potential. That is, we see more than one cell type in a clone. We also see some clones that have only one cell type, for example, just rods. But again, rods are really abundant. Those rod-only clones might come from multipotent progenitors. But we also have evidence for bipotency in that we can see a terminal division of a particular progenitor that makes a cone in a horizontal. Now let's think about those progenitors a bit. So those progenitors, again, are the mitotic cells, and they're making different daughter cell types at different times. So one thing we've wondered is if they're changing over time, and that's what drives the production of these different cell types. So to address this, we did some mixing experiments where you took early progenitors, put them in a late environment, and asked, will they now jump forward and make a lot of rods, bipolars, and Muellers? And the answer was no. They always made the same temporal cohort. We did the reverse, late into an early environment. They did not go back and make early cell types. They still made the late cell types. So this tells us there's intrinsic differences between these two different aged populations. And we think that, in fact, there are intrinsic processes driving this progression. These probably involve transcription factors, chromatin changes, microRNAs, other protein changes. There's a little evidence for transcription factors. Michelle Kayowet showed that early on, Icaros, a transcription factor, gives early properties. And Seth Blackshaw showed Castor, another transcription factor, can give late properties. But there are so many cell types being made, there's got to be a lot more going on. And we hope that we'll be able to figure some of that out. Okay, so how do we explain these changes over time in the progenitor behavior? This is a classic developmental model where the earliest cells have the ability to make all the cell types in the tissue, and they're very proliferative. And over time, they lose that. I don't think that's true. I think instead it's something like this. This is something I proposed more than 20 years ago. And the idea here is that at the beginning, the progenitors are able to make the early cell types, but not the late. They're not competent to make the late. And that's basically demonstrated by moving the early progenitors into the late environment where they don't switch to making late. But we know they have the potential because if we infect early, all of them are making lots of rods and some late cell types. So at that early stage, they're not competent to make the late cell types. And then they also lose it because, again, the late progenitors can't make early cell types. So we think there's a gain and loss of competence, the ability to make cell types in this over time. Okay, so we're still interested not only in this progenitor level, but also in how does that group of progenitors, even at one time, create a lot of diversity. And we decided to tackle this in the bipolar cell system. Now, bipolar cells connect the photoreceptors to the ganglion cells, and there's a reasonable number of them. There's about 15 types, or I should say there is 15 types in the mouse. So we thought that was enough diversity to be interesting, but not so much to overwhelm us. Amacrins, for example, there's 63 types. Now, we wanted to study this in vivo, so we wanted to be able to recognize all of these bipolar types in tissue. And we had some antibodies, and there had been some morphological and physiological descriptions, but we wanted to be able to use probes to recognize each subtype, each of these 15 subtypes, in tissue sections. So for that, we developed RNA FISH probes, fluorescent in situ hybridization probes, based upon single-cell sequencing that we carried out in collaboration with several other groups. Sheldon Rowan, when he was a grad student in the lab, made a GFP line of mice where all the bipolars were green, so we could fax them out and then look at the single-cell RNA profiles, which we did with Aviv Regev, Josh Sains, and Evan McCosco. And from that, we could generate a set of probes, RNA probes, to recognize all of these cells in tissue sections. We used a method called SaberFish for that. We collaborated with Peng Yin's lab to generate this method, SaberFish, which I should say is 10 times cheaper than most of the other methods that you get commercially. And these probes can be designed for specific RNAs, and they can be combined to detect several at the same time. And we can do sequential imaging sessions. So this just shows a pretty picture of a bunch of probes for different retinal cell types. So Emma West and Sylvain Lepan generated 15 probes so that we could recognize all of the bipolar subtypes in tissue. And here's a little bit of their data. So on the left is a WGA outline of retinal cells in a tissue section. In the middle is a reconstructed image of all of those WGA profiles. And on the right, I hope you could see when this slide came up, there were some fluorescent puncta in the bipolar cell layer. That's right in the middle. So Emma worked out a pipeline to outline a cell and to count every FISH puncta across 8 imaging sessions so she could recognize every one of the bipolar subtypes in the tissue section. Now, she used that method to ask several questions. I'm just going to tell you about one of them. We wanted to know where and when are these subtypes generated? And we wanted, again, to do this in vivo and then look in tissue sections and look at when the cells were born. So again, born means the terminal S phase, and we can determine that by applying 2 sequential labels for S phase. One is BRDU, one is EDU. And by looking at BRDU, EDU combinations, we could see if a cell was born before P3. I should say P3 is the peak time of genesis of bipolars in mouse. So we could tell if a cell was born before P3, it would have neither label, or BRDU but no EDU, in which case we knew it was born between P3 and P4. Or if it had the other label from a continuous EDU labeling, it would be born after P4. So we could combine this, the detection of these labels with our FISH probes, and here's what we expected to see. So the x-axis shows the area that we're looking at. So we cut a section along the dorsal ventral axis. And then on the y-axis is the proportion of a given subtype that is born. So I'll just walk you through that. So if you look at the center part of our section and you ask, how many of those, let's say, rod bipolars were born before postnatal day 3, you can see cells that have no S-phase label, and that would be 80% of the rod bipolars in that patch. And as you go more peripherally from that central patch, you'll see fewer and fewer of the rod bipolars were born before P3, meaning they were born after P3, which is the blue curve. So this is what was expected based on all birth dating that we and others had done. And we did see a central to peripheral gradient, but we had quite a surprise when we had this peak and valley pattern of the central to peripheral gradient. Now, we didn't expect this. We hadn't seen this with any other cell type, nor had it been reported, but then no one, including us, had looked with such precision. So this was a real surprise. What I'm showing here is the rod bipolars, but it was true for all the bipolar subtypes, including all 14 cone bipolar types. Now, I asked Emma to estimate how many peak and valleys did she think was in the retina in total, and she estimated there was 500 to 1,000, we're calling them radial units at this moment, so 500 to 1,000 radial units. Okay, so what is the basis of this pattern? How is it formed, and how does it fit with previous existing data? And what dictates the number of these peaks and valleys? So this is a tissue section from Emma showing the BRDU, EDU labels, and again, the central bipolar cells are born before the peripheral ones. And we wondered, might there be signals, maybe pulsatile signals coming from the center, progressing out to the periphery, which induces the different subtypes to form. So Emma did an experiment to ask a question about that. She took out the retina and made whole organ explant cultures. Some of them were left intact and some had the central retina deleted. And then she asked whether the peripheral cells were still mitotic at the same level as in vivo, and did they produce the same repertoire of subtypes? And the answer was it was exactly the same as in vivo without the central retina. So there's no signaling coming from the center that's needed for the peripheral cells to take on their identity. There's something more local going on. So the wave pattern is not formed by these inductive cues progressing from the center. Now I want to talk about some older data that we and others have, and we can now think about that data in light of this pattern. So this is a bit of data we've had for a long time, which I've puzzled over, and I actually love this bit of data. Okay, so what we're seeing is a tissue section from a mouse infected with a lot of virus. So we're looking for dense infections. We thought maybe that would give us some underlying patterns of genesis. And indeed it did. So if you look at the section on the left, there's 4 bipolar clones in a row. So 1, 2, 3, 4. If you look at the far left, it looks like cone bipolar cells with rod siblings. And as you go through the section, we lose the bipolars, and we also don't see Mueller glia. And on the right, we're on the same section going further towards the periphery, and now we see 5 clones that have a Mueller glia plus a bipolar, plus a rod. So that's a hot spot for Mueller glial genesis. In the middle of the retina was a cold spot for both. And it's not because we're looking at the tail end of genesis of those 2 cell types, because all of those cell types are still to be born after P0. And as I told you, they're equally tiled. So there has to be another explanation. So this is the explanation I've come up with. I'm not sure it's correct, but it's the only explanation I've come up with. So what is shown here is a single clone, a rod and an amacrine, from a sparse infection. So most of these infections in the postnatal retina are small and radial. Now we put in a lot of virus, we're sampling additional, let me just explain this. So when the virus goes in, it can't invade the tissue. It's hitting the M phase cells that are sitting at the apical surface. And then after integration, if a cell divides once to make 2 postmitotic daughters, we get a 2-cell clone like the one shown here. When we put in a lot of virus, we're now sampling cells that are closer together. So let's say there were 4 cells at the M phase surface that were sampled by 4 viruses. So those 4 cells were in sync with respect to the cell cycle because they were all in M phase. And then they were in sync with respect to their proliferation capacity because they only went through 1 or 2 more divisions. And they were in sync with respect to the type of cells they made. In this case, 4 that looks like off-cone bipolars. So how could that, what drives that synchrony? It's possible that a very early progenitor, maybe even the earliest, a founder cell, produced daughters which stayed in sync with respect to their proliferation capacity, their cell cycle phase, as well as the kinds of cells that they would make. There's a little evidence for neighbors having the same cell cycle phase, and that's shown here. This is work from Jeff Tremarky. He did in situ hybridization with a probe for a G2 phase marker. And you can see that cells that are right next to each other are in the same phase of the cell cycle, or at least G2. So that is possible then as an explanation for that synchrony of the M phase cells. Now, there's some other older data from lineage analysis from Williams and Goldowitz. They took 2 genotypes of mice, mixed them together as a blastocyst, implanted them, and then analyzed the mature pups. And you can tell one genotype from the other with histochemical stains. So you see these big blocks of a single genotype. Again, they're radial, just like the retroviral clones. They're much bigger because the marking started much earlier in development. So they interpreted these as founder cell clones, and I would agree. And they estimated there's 500 to 1,000 founder cells, which is the same number that Emma estimated from her work. And 500 is an interesting number because that's the number of the wide field amacrines. Okay, so to get back to the questions then, how does the peak and valley wave pattern fit with existing data? Well, the retroviral clones in the chimeric mice show some suggestive clonal patterns that might match up with a peak and valley. And what dictates the number of peaks and valleys? Well, maybe it's the founder cell number. Again, from 2 independent ways of looking at it, we have the same founder cell number. So what could explain all of these findings? So again, we have these big blocks of radial clones, we have these peaks and valleys, and we have these hot spots and cold spots. So Emma West joined forces with Chang Healy, a postdoc in Cliff Tabin's lab next door, and they came up with a hierarchical model that actually fits all this data. And they ran a simulation of the model, and it actually fits the data. So the idea is that the very early retina, they're founder cells. These are the cells that are dividing to make a bigger retina. So over time, we get a bigger neuroepithelium, but everybody in the retina is still mitotic. Then there's a period of neurogenesis that occurs, starting from the center, progressing to the periphery. And the key part of the model is this slide. Okay, so the founder cells initially are making more founder cells. They then switch to an asymmetric division to make what we call a neurogenic progenitor and another founder cell. Now that neurogenic progenitor takes on the ability to start making the neurons. In the first case, it would make ganglion cells. And that neurogenic progenitor goes through progressions, and its daughters go through progressions in a single direction to change the competence to make the different types of neurons that we see over time generated in the retina. Now, I want to explain something about the competence window. It is the ability to make a cell type. It doesn't mean a progenitor cell will make that cell type. It might need another hit, and perhaps an environmental influence. There might be some stochasticity. Sometimes it does, sometimes it doesn't. It also could be that we can adjust the number of each cell type that's made by making the competence windows big, long, or short. For example, those wide field amacrins, I told you there's only 500. And if founder cells are really truly 500, then every one of those big blocks automatically is going to have one amacrin cell if the competence window to make one of those amacrin cells is really short, let's say 10 minutes. Just probabilistically, it might be that one of those big founder cell clones doesn't generate more than one wide field amacrin, and so on. You can also adjust this in terms of evolution by lengthening or shortening a competence window or adding some new ones. So in this model then, this subclone that's now going through a neurogenic process gets to the end of development to bipolar making time, and in that radial unit, that big radial unit, some of these subclones will be already making the firstborn of the subtypes. But meanwhile, there's more subclones being made, and this continues so that in the founder cell clone, we now have a lot of subclones, and each subclone is offset from the others by a single cell cycle. And all the subclones stay radial. So just using those ideas, they made a model in the computer that matches our real data. So on the left is the model prediction, and it gives peaks and valleys for the reasons I just described. And on the right is the real data. Now, we don't know if this is true. Brian Delgado in the lab is making a much more sophisticated lineage tracing method using Prime Editor, where we will be able to track every cell division in a clone and ask what daughters does that clone make at each division. And the model we have has strong predictions as to how that should look. So hopefully we'll be able to test this model directly. Okay, I'm going to switch now and talk about the formation of a high acuity area. So again, we have our human fovea as our most well-known, but other animals also have them. And I'll be able to talk a little bit about those. So we started studying this in the chicken, the reason being that most mammals don't have a high acuity area, only some primates do. We study rats and mice, and they don't have it. But you probably know that birds of prey have not only one, but two high acuity areas, two foveas, and some lizards have two, other animals have one. And we discovered about 20 years ago that the chicken has one. So shown down here is the people over the years that have worked on this project. It started with Suzanne Brune. And currently here, Joysher, Isabella Vanderweed, and Monica Ayton are studying this process in the lab. And on the left is a flat mount of a chicken retina early in development showing some interesting patterns. Okay, so the first pattern we saw was that the chicken has a rod-free zone and a rod-sparse horizontal streak. This was done by in situ hybridization for rhodopsin. This is near the end of chicken development. And so we were very intrigued by this pattern, and we thought we might be able to study the development of the pattern. Again, you can see in the high mags on the right that there's a nice rod-free zone there, characteristic of the fovea. And it's nice that Curcia showed her images. I was hoping she would. So we can also see that the cells, the cone cells in the middle of that rod-free zone have the same size and shape as the human ones. So again, you can see Christine's human cones. They're skinny, they're packed in, they're very regular in the center, and then outside in the periphery, the cones are much bigger. The chicken has the same diameter of the inner segment and is packed just as densely as in the human fovea. And again, the periphery has wider intersegment diameters. So that's two features, rod-free, and it has this dense packing and skinny cones. So we started looking then how these patterns might form in the chicken, and we had in situ probes for a number of genes, either from the literature or discovered in our lab, and that's what I'm showing you here. So on the top left, the red stain is the RNA for a synthetic enzyme for retinoic acid. On the bottom is the pink is another synthetic enzyme for retinoic acid. And then we discovered this beautiful green spot and stripe. Here, Joycer showed that this spot and stripe, this FGF8, is in exactly the region throughout development where the high acuity area will develop. I should also say we looked at ganglion cell density, and it's 2 to 3 times denser in the area with no rods, and the internuclear layer also has an interesting structure called the aster, and all of those line up in what we think is a high acuity area. So FGF8 marks that from the very beginning of development, and it persists all the way through. The top right shows some interesting patterns for the destruction of retinoic acid, CYP26A1 and CYP26C1. C1 is that bright blue spot which sits right on top of the FGF8. So those destroy retinoic acid signaling, and Susanna Da Silva, when she was in the lab, showed that FGF8 and retinoic acid signaling were important to form this rod-free zone and high ganglion density area. So they're not just pretty pictures, but they're actually functionally important. There's some other nice images shown there. We now have quite a few genes that show up in these interesting patterns. We call them our dorsal ventral, anterior, posterior, spot and stripe, and then we also have a vertical stripe, which I'm not showing. So a lot of beautiful patterns in the chicken. So we wondered, did these patterns also occur in other species like humans? Here, oh, sorry, I just wanted to show you this cute little representation of some of the animals we're working on. So human, they're in the center. Zebra finches have 2 high acuity areas. So do anoles, they're the lizard. And then other birds either weren't described or had a single fovea. So we decided to look with the probes for those pattern genes from chicken in other species. So Susanna looked at human fetuses. This is about a week 11 human fetal eye. And on the top right, you can see the synthetic enzymes for retinoic acid. There's one in the dorsal and one in the ventral, just like in the chicken. And then the bottom is the CYP26A1, the destructive enzyme, in a restricted pattern. If Susanna hadn't cut it right there, it might look a little bit more like it's in the right position, but we think that that is marking the future fovea. Now, that was tedious to go through a lot of in situ hybridizations, and we were wondering, could we use single-cell RNA data, there's now quite an abundance of it, to also find out about patterning genes in other species. And so we joined forces with Chang-Hee Lee using data that was gathered by Nicholas Lonfot when he was in the lab of single-cell profiles from chick retina at this time in development. And what Chang-Hee figured out how to do was to look at every single cell and ask, how much does it have of this dorsal marker or this ventral marker or this anterior or posterior marker? And just by evaluating 6 genes that mark those coordinates, he could then place that cell in a location on a 2D map. So on the top, I'm showing you the real in situ data for chicken probes, and then those 3 images that are blue and green on the bottom are the reconstructed 2D maps. Every dot is a cell that was evaluated for the expression level of those patterning genes, and then it was placed accordingly. So you can see this method recapitulates the real data quite well. This is for dorsal ventral genes. We also have it for anterior posterior genes, and again, they're beautifully patterned using this 2D map reconstruction. So these data are all from the chick, but we had a lot of human data, principally from Botan Raska's lab. Botan and his crew had evaluated both human fetal development as well as organoid development, including these early times. So by looking at those profiles, we could see if human data also matched, and you can see that it matches pretty well for these different genes. The dorsal ventral and the anterior posterior genes are nicely patterned. Those are all the images on the bottom. FGF8 has a bit of a strange pattern. We're not sure what that one means. But this means that we can look at any species where there's single-cell RNA data and ask about these patterning genes. We can also use it to discover new patterned genes. It will also direct us as to which genes do we want to use on more precious tissue. Okay, so we wanted to look also at other species, and this is just a compilation of some birds, chicken, duck, quail, and zebrafinch, looking at FGF8 and the retinoic acid synthetic enzymes, and you can see they all have the same pattern. We are also looking at adults, and we're quantifying the cones and the rods to see how that plays out in the final tissue. We looked also at the degradative enzymes, the CYP26A1 and C1. On the top is the chicken, and on the bottom is the duck, quail, and zebrafinch. And you can see that we have these nice spots of CYP26C1, just like in the chicken, and a stripe of the CYP26A1. Now, we were excited to go a little bit beyond birds and look at lizards, because they're further away evolutionarily, and we looked at the brown anoles, which has two foveas. And so on the left are sections from the chicken showing the retinoic acid synthetic enzymes. And then the middle, the sort of orangish and blue, is from the anole. And they have the same patterns, dorsal and ventral, for the synthetic enzymes. And on the right are the FGF8 and CYP26C1 patterns, again for chicken and anole. And it looks like they're in the right place to, again, be playing the same functional role in the brown anoles. So to summarize what we've done then across species, in birds, lizards, and mammals, we have evidence for conservation of expression of these patterning genes. And we're interested in going further, looking at fish to see if a common ancestor was using the same patterning genes. And we're also looking in the adults. We can now do in situ hybridizations for all of the opsins and looking at how the various opsins are arranged in these different species. So we've been fortunate to have great funding over the years. As was mentioned, HHMI has been fantastic for us, National Eye Institute as well. And we've also had good funding from other sources. And I'll finish up just showing you a picture of my current lab. So thank you very much for all being here.